Hi, everyone, and thanks for making time on this really lovely overcast Friday evening. And the weekend is about to start, so I think this is a really good way to kind of close the week. Well, as you know, the Inside Out Human Body series is an outreach initiative from the Bangalore Life Science Cluster. The cluster comprises 3 research institutes. There is the National Center for Biological Science that specializes in fundamental research. And there is the Institute for Stem Cell Science and Regenerative Medicine, which is inStem, and that focuses on translational research. And they are backed by CCAMP, which is the Center for Cellular and Molecular Platforms. And CCAMP is this really great incubation hub for startups that do biotech. So we found that this kind of initiative is really important to kind of introduce people to the kind of science that is out there and also the kind of science that we do. And this is an initiative of the Communications Office of the Bangalore Life Science Cluster. So without any further ado, I'm going to lay this stage open for Raj to introduce our speaker. Take it away, Raj.
Thanks, Maheen, and welcome everyone to the second talk of this series. Today we're very happy to introduce you to Bhavana, who is a faculty member at inStem. She is a DBT Wellcome Trust fellow. She's been in inStem for 2 years, 1 year, 2 years. She 1 year. Okay, so she's fresh from her research in London, and before that she was working in TIFR Mumbai. She has been working on the cortex, the seat of higher brain function in humans, and today she's going to talk about the brain. So Bhavana, thank you very much for doing this. Please take it away.
Okay, so just before Bhavana begins, we've got a couple of questions which we'd like to ask you all. So I'll just begin with those questions right now. Excuse me for a minute. Okay. Yes. So this is our first question. And what is more, the number of neurons in the human brain or the number of people on Earth? Which is more? And we're just launching the poll. You guys can vote. We'll just keep it open for a few minutes.
I think it's really unfair, Chandrakant, that we're not allowed to vote.
Oh,
that just sucks.
We can do that for the next question.
Okay, great, because I really want to know.
Okay. So we've had 70% voting. Come on, guys, let's do this. Okay, we've got 15 more seconds. People on YouTube, yeah, you can put your answer in the comments, and okay, so I'm ending the poll now. Let's just look at the results. So 85% of you said that the number of neurons in the human brain are more than the number of people on Earth. Bhavana, would you like to answer that question?
Yes, yes. All those 85% of people are right. But what really would surprise you is that it's the number of neurons in the human brain are about 86 billion, and the number of people on Earth are 7.8 billion. That's more than 10 times the number of people on Earth, the number of neurons which are sitting in your brain right now.
Wow, that's pretty amazing. And well, we've got we're gonna go to our next question. Well, if guys, those of you who got it wrong, just think about the number of neurons that it took to get that wrong. So there is, what's the simplest organism with a brain? So you got the images there. And our second question is that, what is the simplest organism with a brain? Yeah. So is it Hydra, a freshwater organism? Okay. Is it the roundworm, C. elegans, or the fruit fly? So yeah, well, not as this is a tricky one. Okay. Just I'm going to keep it open for another 20 seconds. People on YouTube, please comment. Which one is it? Is it Hydra? Is it roundworm? Is it fruit fly? Imagine the KBC music at the back, although you're not going to get any money for this. That's fine. When I think next time around, we'll also have some music as well for this. So, okay, that's I'm going to end that poll, and we will look at what did you all say? So 42% said Hydra, roundworm got 33%, and Drosophila got 25%. So it's not as completely one-sided as the last one. So I guess Bhavana can answer
Yes.
this one as well. Which one is it?
I think people chose their favorite model organism and voted for it. So the answer is Hydra. It's the simplest organism with a brain, and even with a few thousand cells, that it has some sort of a structure like a brain with which it perceives the world around it.
Awesome,
So
awesome.
Hydra.
It's Hydra. Well, I'm glad it's Hydra because I actually worked on Hydra for 3 years in my college thing. So anyways, so that's our poll. Thank you guys for participating in this. And we will, once now the fun is over, well, it's just beginning actually, we'll go over to Bhavana, and Bhavana can begin her talk. Over to you.
Okay. Thank you, Chandrakant. Thank you, Mahin. And thank you, Raj, for this nice introduction. I will share my screen. So, yes. So the sweet smell of this rose, which I've put up as a picture here, the sweet smell of this rose or this colorful butterfly that you see, both these experiences you cannot have if this 1.5 kilogram tissue mass, which is the brain, which sits on the top of your head. So what I today want to convey in my talk is how this structure comes, what are the fundamental features of this structure called the brain, how this brain develops during development, and finally, how this structure comes about to do its function, and that of the perception of the world around you. But to come to this understanding that the brain is the seat of intelligence, it took about 5,000 years. The ancient Egyptians, about 5,000 years ago, who were keeping mummies, they kept the heart because they thought the heart is the seat of intelligence and not the brain, and they completely removed the brain. And this thought persisted for 1700 odd more years, when the very famous philosopher Aristotle thought that the brain is just a radiator to keep the heart from overheating. His contemporary, Hippocrates, who's considered the father of medicine, he stated that the brain is the seat of intelligence, and that the brain could be attributed for all the sensations we have of the world. But not until the 1900s did we come about the very modern thought process about the brain and the modern neuroscience. This was revolutionized by a scientist, Santiago Ramon Cajal, who was an anatomist, and he's considered the father of modern neuroscience. He proposed the neuron doctrine, where he suggested that the neurons, which are the building blocks of the brain, they are the fundamental way by which the brain functions. He was able to do this because of the advent of microscopy and staining technologies. He painstakingly drew with hand the neurons from several different regions of the brain and from several different organisms. And some of his images, as the one which you can see here, are so beautiful and so intricately detailed that they are even used today by medical students and scientists. He further emphasized the connectivity of neurons to be important for brain function. He said that the neurons which connect with one another, that is the fundamental basis by which neurons function, and that leads to the brain function. And for this fundamental discovery and for this study, he received the Nobel Prize in 1906, along with Camillo Golgi, who gave his very revolutionary method of staining. And this is Raman here shown in his laboratory with some of the very primitive microscopes and working his way on some of these sections of the brain and drawing them on his paper. Right. So in my talk today, what I'm going to tell you what are some of the fundamental features of the brain and how these features come about during development, because the way these features come about or the way the structure is made, that determines its function. And finally, tell you about one such function of the brain of how it perceives the world by telling you or giving you examples from one sensory modality which we use to perceive the world, that is the color vision. Okay, so let's begin with understanding what are the fundamental features of the brain. Before we go into what the brain is, the nervous system is actually something which encompasses both the brain and it encompasses everything which has got to do with nerves or neurons in the body. So simply put, it is the sum total of all the nerves, connections in the body that send or receive signals all over the body. And as I said, brain or the spinal cord is a part of it. It consists of two parts. First is the central nervous system and the other is the peripheral nervous system. And for my talk today, I'll be focusing on the central nervous system, which consists of the brain and the spinal cord. But what the peripheral nervous system does is all those neurons which are in all over the body, it helps these neurons to connect with the brain and the spinal cord. So in a way, it helps relate the information which comes from the brain and to take it to the different parts of the body, and similarly getting information from different parts of the body to the brain and the spinal cord. Now, before I go and describe to you the structure of the brain or some of the fundamental features of the brain, what I would like to tell you here is, because this is about anatomy, it is important for us to know that how or which plane are we talking about when we're looking at a structure. And the brain being a 3D structure, it becomes important to understand that at which plane was the section cut for us to understand what information is giving. Nonetheless, any plane you cut, the brain is full and rich of information at every plane, and every region of the brain has some information to give. So in this talk today, I'll be talking to you about 3 planes of sectioning. First is the horizontal. As you can see here, it is a sectioning which is cut across the horizontal plane. And what information you get from such a section is, as you can see here, this is a section which is cut horizontally. Or you can say that this is a top view of the brain. Here is the, wherever my pointer is there is a nose. You can see the eye sockets, and you can see this is the front of the brain, this is the back of the brain. And you can see this region here is the cerebral cortex, or the seat in the brain where all your higher order functions happen. Now, if you would cut face-wise, that is, you cut your sections like this, what you would see is like a section like this, where you can actually see the spinal cord here. You can again see the cerebral cortex and here you can very well appreciate the fact that the cerebral cortex has folded into itself. Can you see some of these gray regions and black regions where the cerebral cortex has folded into itself? And then comes the sagittal plane, or the side view, which and here you can actually see the entire brain and the different parts of the brain. And here is the nose, but this is the brain and this is the cerebral cortex. But you can see other regions of the brain which are important for several different functions, some of which I'll be coming to just in a slide or two. Here is the cerebral cortex, here is the brainstem, and going on to the spinal cord, right? So what is the structure of the brain? I think some of you might have Googled and would have seen that the brain looks like this. And the prominent things which are immediately visible as soon as you look at the 3D structure of the brain is this numerous gyri and sulci, or as you may call them, the crest and the troughs. These are simply existing in the brain because the brain, human cerebral cortex is so huge that to fit it into the skull, the structure had to fold in itself to fit it into that skull, which is on top of our head. Now, as I told you, this is a side view, and you can see the cerebral cortex here. But along with that, the moment you expose and cut a section, not only do you see the cerebral cortex, but you start seeing other structures, namely the cerebellum here, and hypothalamus and all of that. For the purposes of my talk today, I'll be telling you more about the cerebral cortex, but just briefly touch upon what are some of these other structures which exist in the brain. Some of these other structures are cerebellum, the hypothalamus, the pituitary gland, or the medulla oblongata. And these perform some of the, very important functions. So in the very first, I told you that one of the functions which the brain does is perception, that is looking at a flower or, smelling of a rose, but it also controls a lot of bodily functions. And some of these functions are unconscious. For instance, the sense of balance which you get comes from the cerebellum. Whereas, the body temperature is regulated by the hypothalamus. And the structure here, which is the pineal gland, that is very important for regulating sleep. So these are some of the other bodily functions which the brain carries out. But I think for today's talk, I would like to really focus on one of these higher order functions, which is your thinking, memory, perception, language, or and all of that, and finally even consciousness, and all of that comes from this part of the brain, which is the cerebral cortex or the cerebrum. Right. So as I said, that the cerebral cortex is the seat of higher functions, and it does all these complex functions like learning, memory, and emotions. This cerebral cortex is further divided into several lobes, and each function is further divided into specific parts of this cerebral cortex. For instance, the frontal lobe is involved in your motor control, that is control of movement. It is also important for language. It is important for thinking. It is important for making decisions. I mean, one of the main regions which is involved in decision making is this frontal lobe. Whereas the parietal lobe, and I think this will become very clear in this next schematic here, that the parietal lobe is involved in all your perception of the touch senses. The occipital lobe is being devoted for the vision. So all your vision related things happen in the occipital lobe. Whereas the audition or the hearing, the sense, the region of the brain which processes the auditory modality, auditory sensory modality or hearing is the temporal lobe. Right. Now, up till now, I told you about the overall structure of the brain, that, what are the different regions or even what, each region does. But one very important thing is, finally, when we talk about function in a nervous system, or in this case, brain or the cerebral cortex, it is important for us to understand that it is an emerging function which comes basically from the cells which are at the core of all of this. And the two major types of cells which the brain contains are the neurons and the glia. And in my next slide, I'll be telling you about the functions of each of this. But once these neurons and glia are present in the brain, what they do is that the neurons, they connect with each other to form some sort of a circuit, which then gives rise to a physiology and then gives rise to a function, which is perception. Because the brain is all about transmission of signal, and that this property comes from its cells, following by the formation of connections or formation of the circuits, leading to a certain physiology in those cells, leading to a function where in this case could be perception, which could be the 5 senses. And based on how we perceive the world, it can also lead to behaviors or how we behave in the world. So as I was telling you, the fundamental thing in the nervous system is the cells, and this is a theory which has been which was initially proposed by Carl, and even today it still stands the test of time. In fact, what he proposed in the 1900s, some of these proofs came only by 1960s and 70s, and we're still generating proofs or still trying to understand how the properties or how the cells finally lead to the function of the brain. So as I said, the fundamental unit in the brain is the neuron, which is the functional unit, or the one cell which fires, which conducts the electrical signal. So since this is a very important function of the neuron, the structure also has to be in such a way that it helps or facilitates in conducting such a signal, an electrical signal. So what a neuron has is that it has a cell body, which consists of several dendrites. So whenever an electrical signal is generated upstream of it, it is able to receive the electrical signal via these dendrites onto this. And then it has a long, protrusion here, which is called the axon, with which it is able to propagate the signal to the downstream cell or a downstream neuron by which it is connected. So basically, the signal which it gets as an input, it's able to give out a signal as an output via its axon terminals to a downstream neuron. And that is the fundamental way by which signal or electrical signal or connectivity happens in the brain, is via the neurons, the formation of the axons, and how they connect to the downstream, downstream neuron. Now, this is just not enough for a functional brain, because once you have a functional cell, you also need a support cell. And that cell is the glia. And for a very long time, I would perhaps say even 20 years before, the glial cells in the brain, which actually are 1 trillion, they are 10 times more than the number of neurons, which that is, if the human brain has 86 billion neurons, then it has 1 trillion glial cells in the brain. So these are the support cells, and they support the function of the neurons in every possible way. And I've highlighted here some of the glial cells, the astrocytes, the oligodendrocytes, and the microglia, which help the neuron in performing its function. And therefore, they are the support cells in the brain. Right. So till now, I've been telling you about what are the fundamental features of the brain. I hope, you understood that, what starts at the cell makes a connection, leads to physiology or to a tissue structure, which then, of course, there are several regions in the brain which do what function they do. But I think it's important here to pause and understand that this structure this was just not like, you took bricks and built the brain. So this structure came about during development, or when we were all during gestation in the mother's womb. This brain develops, as does several different organs. And the way this brain develops is very important to determine its structure in adulthood. So in the second part of my talk, I'm going to be talking about how the brain develops and also talk a bit about my own work in understanding how the cerebral cortex develops. And if some of you, if in the audience there are some school students, and if you're really getting bored with all these technical terms like structures and function and all of that, I would suggest just wait for the third part of my talk about perception, because I'm going to be talking about some illusions or showing you some illusions, and that will be fun. Right. So even before I go to talk about how the human brain develops, I would like to show you this video so that you appreciate that what was even smaller than a 1 cent goes on to become this huge 1.5 kilogram mass. And also what starts with a few number of cells generates this, brain structured with trillions of cells. And that's the thing which I would like to appreciate. I'm going to start this video.
So we're introducing ourselves into this process about 1 month after fertilization, when the entire embryo is smaller than the size of a dime. And we're going to look in at the nervous system where relatively small numbers of cells have been allocated, and then monitor over the 9 months of human gestation the increase in the number of cells and the change in the shape of the central nervous system. So by 3 months, we have something like 1 million cells. The nervous system is beginning to exceed the size of the dime. You can see that the future forebrain and the cortical region are expanding disproportionately to the rest of the nervous system. By 6 months, we have hundreds of millions of neurons, and the brain is beginning to take now a familiar shape. You can see the beginnings of the indentations, the sulci and gyri that Eric described, so that by the time we approach the 8th and 9 months, this is now a familiar picture, and we have tens of billions of nerve cells present. So we're introducing ourselves into this process about 1 month after fertilization.
Okay. Right. So, so as I told you, why I really wanted you to see this video was to appreciate the fact that, you know, why it takes 9 months for this entire structure to come about. Right. So a few slides back, I spoke to you about the emerging function in the nervous system, like how the cells come together, they form connections, and then that leads to the function. But now I'm going to tell you that this is the very same way in which the brain also develops, and that's how this entire structure-function relationship comes about. So, so the building of the neural network also consists of similar steps. For instance, it starts with a pool of cells called stem cells, which go on to generate the entire brain. Yes, the entire brain, which consists of trillions of cells, comes from those few thousands of stem cells, which keep dividing and generating neurons and glia and the different cell types in the brain. So the stem cells divide, they generate the neurons and the glia. So basically these neurons all, which are the structural or the functional or the basic unit in the brain, they all come from these stem cells. These neurons and glia, as they develop, they also give out projections. The neurons are generated and they send out their long axon projections. And these axon projections connect with other neurons. And that is a very important process which happens during development for these connections to come about in adulthood. A small mistake in any of these steps, right from whether, a mistake in the stem cell not dividing properly, you will actually end up having a very small brain. That's a condition called microcephaly. If you don't have the right number of neurons, you could have neurodevelopmental disorders like autism. If you don't have, the right connections, in fact, if you have aberrant connections, there is a condition called synesthesia, which I'll be talking about towards the end of my talk, which happens because if the connections don't happen well. So each of this step is very, very crucial, and each of this step is highly regulated so that you have a functional brain, which does, you have a functional network and a functional brain, which does its function of both physiology and, for instance, perception and behavior. So as I was telling, it is important that the stem cell divides and makes these neurons. And in the developing brain, how this happens is that the stem cell divides, and as it divides, one cell becomes a stem cell, so that there is a pool of stem cells which continue to keep dividing and thus making the entire brain, whereas the other cell begins to differentiate into this neuron, and that is how different neurons in the brain are generated. Now I'm going to be telling you about my own work in this, that how we studied the mouse brain to understand how this process happens in the mouse brain, that how this stem cell gives rise to a particular class of neurons. Just so that, I'll be showing you the mouse brain, and so that you don't get confused, I would like to say here that though several features of the human, the fundamental features or features which are very important to make this brain are quite conserved between the human brain and the mouse brain, there are several differences. And one of them, which you may immediately see, is that the human brain consists of this sulci and gyri, which is totally absent from the mouse brain, simply because it is not that as huge as the human brain. And so it is a very smooth as compared to the human brain. So as I was telling you, that this pool of stem cells, which make the entire neurons, they all reside in a zone called the cortical primordium. And this is called a primordium because it's still not the adult functional cerebral cortex. It is still a primordium. This is full of stem cells. So if you cut a coronal view, a face view, you would see that this cortex, which is developing, consists of this yellow zone, which is full of stem cells. And at this stage, only a thin sliver of neurons exists at this stage. So this is the developing cortical primordium. As I said, all the entire region is full of stem cells, whereas this is just a small amount of neurons. Now this structure, in case of mouse, it only takes 20 days for it to develop. So within a span of 20 days, it has to produce this cerebral cortex, which has about 6 different layers of neurons, each doing different function. So how does this come about? This comes about by a series of cell divisions, as I told you, but which are highly regulated and highly, regulated in a stepwise manner. So this is the pool of stem cells which I was talking about, which begins this process of generation of the cerebral cortex. The very first day in embryonic development. So very first day in embryonic development, which we call as embryonic day 11.5. So just to give you a context, the entire mouse develops in about 20 days. So if 0 is the time point at which one egg and sperm fused, at about day 11 in the developing in a pregnant mouse, at about day 11, this is what is happening inside the brain of a mouse. So it contains stem cells, and the very first day when these stem cells divide by the division, which I told you, where it gives rise to one stem cell and gives rise to one neuron, the very first day it gives rise to a kind of neurons which are called layer 6 neurons. This happens at E11.5. Now at this stage, I mean, just so that you can can understand this better, I have here put a brain where this is being marked by a protein called TBR1, and this is how layer 6 neurons look, the blue, which is at the bottom of this brain. The very next day, this very same stem cells, now instead of producing layer 6 neurons, they switch and they start producing another set of neurons, which are called layer 5 neurons. And here, as you can see, they are marked by the protein called FESF2. So there is layer 6, on top of which there is layer 5. So what happens is the neurons are produced here, they migrate past layer 6 and they go on and sit at layer 5. Now, every day in mouse development, this keeps happening. So on day 13, what happens is the layer 4 neurons are generated, which is marked here by a protein called ROR beta. And by embryonic day 15, you generate all the 6 layers of the cortex here. And this is CUX2, which marks both layer 4 and layer 2-3. So as you can see that this is a highly regulated process, like a question which we asked, that if suppose we perturb this, if suppose we perturb it at say embryonic day 12 or 11, by removing a certain factor which could be important for making these neurons, what could happen? Can we disturb this process? Can we end up with a brain which could have slightly more number of neurons of one kind as opposed to the other? Turns out the answer is yes. This is work which I did. Sorry. Yes, sorry, it's here. So as you can see here, this I'm showing you a brain, and we call this as a control brain. Why we call this as a control brain is the brain where we have now not done any perturbation, like we've not gone ahead and removed some protein or some molecule. So here you can see that this is layer 6 marked here by TBR1. And this is layer 5 marked by 3 different markers. FESF2, I showed you a few slides back. Again, ER81 and marked by IT2 as well. Now, if we remove a certain molecule called LHX2, what we found to our surprise that, this layer 6 was reduced and this layer 5 was expanded or increased. And a fact that you can appreciate well here, which is marked or stained with a different marker. This is control here, and this is this is control here, and this is the removal of the molecule called LHX2. And you can immediately see there are two things which happened to this brain. One, that this brain shrunk or decreased in size. On two, along with the decrease in size, we saw a reduction in this layer 6 neurons along with an increase in layer 5 neurons. And this is just a bar graph for you to understand that just not by visual, but even by numbers, we see an increase in this neurons of the layer 5 and a decrease in the neurons of the layer 6. Now, why is this important, right? So as I was telling you that the numbers of neurons in each and every layer is important for its function, because it's the neurons which connect and ultimately decide the function. So here, as you can see, that this layer 6 neurons, this is the mouse cortex, so it is not going to be folded, it's going to be smooth. And this is the layer, and this is a sagittal view. I'm sure you now appreciate and understand that this, I'm showing you a side view of the brain here now. And this is the layer 6 here. And as you can see that the layer 6 goes on to connect to a region in the brain, which is involved in, it's a relay center. It's a center where it integrates all the information from your sensory modalities, which means like things which are coming from your touch, coming from your hearing or vision, they all come to this center. And then from the center, they go to the brain. Similarly, from the brain, neurons come from the brain, which is this part of the brain, and they go to the, the thalamus. So the layer 6 neurons, they connect or they give out projections which go to this relay center. But the layer 5 neurons do a different job. They connect to the spinal cord, and they consist of the motor, cerebrospinal. They consist of the upper motor neurons, which connect the brain to the spinal cord and help you in your movement. So as you can appreciate, because the neurons determine the function or the connectivity, it is important during development to tightly regulate the process. And one mistake or a mutation in the protein like LHX2 could end up in a cortex, which will have more number of neurons of the thalamus and less number of neurons which project to the spinal cord. So thus, it is important that the brain develops, and the brain develops well, so that the fundamental features of the brain are intact, and this structure then goes on to do its function. So that brings me to the last part of my talk, which is the perception. And before I even begin, I should tell you that I'm a developmental neurobiologist, and that what I'm going to talk to you about is all cognitive neuroscience. I'm not an expert in cognitive neuroscience, but from the papers which I've read, and also from the talks which I've attended from the people who've worked in this field, I've put together this part of my talk to really bring about the fact that, how, the process of, perception happens in the brain. And just to show you with the example from the color, how the color vision happens. Right. So coming to perception, and that is just one of the functions of the brain. There's several functions which it does, like language, memory, and everything. But I'm taking this example just to drive home the point that, perception happens in the brain. Okay. So how does the human eye sense color? So as we all know, we have the human eye with which we see the world, and this is the organ which we have with which we also see the colorful world around us. And a quick look into the eye will tell you how this visual perception starts in the eye. So if this is a pencil here, light signals from this pencil, they are converged by this lens, and many of you would be physics students and will appreciate that this image is formed on the retina. So the retina is the screen in the eye where the image is formed. So till now, it is all physics, because till now it is light, which comes from the pencil and which forms an image on the retina of the eye. Now, what happens after this? So what happens is, the first step is this light is converted to a signal, that is an electrical signal, and that happens in this region of the retina, and I'll come to that in a bit. And once that signal is converted from light to electrical signal, that signal then goes to the brain and it is interpreted. How it is interpreted? So first, looking into the retina, what happens is the light which comes from this pencil, which comes here, the retina has a series of photoreceptors called rods and cones. Cones are the ones which are involved for color vision. Rods, they look at brightness. So this retina is full of these photoreceptors. And when we, so as I was saying, we consist of, the eye has two kinds of photoreceptors, namely the rods and the cones. And the cones do the color vision, whereas the rods are involved in looking at brightness. What happens in the receptor, photoreceptor? And this is a very detailed, picture of this photoreceptor, which consists of, several different things. But just to give you a brief idea of what happens inside this receptor is that the light which comes from the outside world is converted by a visual pigment. Now, I must mention here that this visual pigment is made from the vitamin A. And that's why it's very important that we consume a lot of carrots, because it's important for vision. So this visual pigment then converts, relays this signal to some downstream protein, which then leads to an influx of ions of sodium. And because sodium ion rushes inside the cell, there is a sort of an imbalance created between the inside and outside of the cell, and that leads to creation of a potential gradient, or that is how an electrical signal is generated. So basically, it is the photoreceptors of the eye which convert the light into the electrical signal. Fine. So now the light has become an electrical signal. Now what? Now the signal goes to the brain, and here is what the brain does. The brain extracts information from this signal. So from the retina, your electrical signal through many different ways goes to the visual cortex. So as I told you before, the occipital lobe is the region where the vision happens. This signal directly goes to the primary areas of the visual cortex, where it is split into two streams. One stream asks the question, what about this world which I'm seeing? For instance, the pencil, what is the color of the pencil? Or what is the form of the pencil? That bit of information is processed in an entirely different region of the brain. And the second information, that about distance or movement, or the how or the where pathway, that is processed in a different region in the cortex. So, if you can appreciate that the information which came as a single unit into the retina has now been split into two different parts, asking the questions about color, form, asking the questions about distance and movement. These will then be separately processed in this higher center in the brain. This is the frontal cortex, where it will, match it to your own memory of a pencil. For instance, you may have seen a pencil, a same color pencil lying somewhere, or that you may have heard your teacher saying that this is a pencil and this is how it looks. So it is taking all the information which is gleaned from your hearing or gleaned from seeing, all of that, it will then match with the information which came from these two different pathways, integrated, understand that it's a pencil, and then tell you, okay, now you decide, now let's decide whether we need to pick up the pencil or not. And you will pick up the pencil if you really want to write. So just to drive home the point again, what was there in the outer world was this signal, that is the light signal, that this is the visual world which has the signal, but the brain has extracted information of color, of orientation, of size and distance, and it has made a feature maps of these. It has then taken it to its own master map, where it is now, it perhaps, depending on whether you want to write, you may have the preference for writing with a yellow color pencil. You may want to decide based on information from the color and based on how far the pencil is, distance, whether you really want to go and pick it up, this master map, or the higher region of the brain, which is the frontal cortex, decides or focuses its attention, decides what it needs to do about the visual information. So what I really want to emphasize here is, from this hypothetical model, is that light may come from the visual world, but it's broken down into these essential features by the brain, where it takes all this information and decides what to do. And all of this happens in fractions of a second that you don't even know that this is constantly happening in your brain all the time. Every time you see the world, you interact with it, you decide to do something. And that to me is like, a fascinating concept, which I thought I'll share with you. Now, just to drive home the point a little bit more, I have some illusions for you just to say how the brain perceives the information. So if you look at these two squares, which is on the screen, and if I ask you the question that which shade of blue is darker, the one on the left or one on the right, I'm sure all of you will agree with me if I say the shade of blue on the left, that is left for me, is darker than the shade of blue which is on this side of the screen, right? Now, if I say, let's check this by joining the two shades. And as you can see, the two shades are of the same shade of blue, right? So what has happened here? What has happened here is that your brain just not sees this blue color as a standalone blue color, but it perceives this blue color along with the, with this boundary around this, which boundary around it. Similarly, for this blue color also, it uses the boundary around this to perceive, to look at the color in its entirety. And because of this perception, we see that, and this is, and what is the principle behind it? What is happening is, once this cones here, so every cone maps to a neuron, and that's how it relays the information. Once a cone here perceives the light signal, what it does, it also, some of these neurons which are downstream of the cone, they could also inhibit laterally. For instance, if this cell was firing, what it immediately does, it does not allow these neurons to fire next to each other. That is called lateral inhibition. And this is one of the principles by which brain perceives not just the color which is coming or the light which is coming from a single point in the visual field, but also in its background. So what is actually happening? So this is just my interpretation of it. This is my simplistic interpretation of this. Things are much more complex than this, but this is how I think your brain sees this to be a shade darker than this. What is happening is that cones which, in your retina, which look at this color, cones look at this color, because this is a light shade, it is firing, say, at +10, and simultaneously inhibiting this neuron here by -10. So this seems like much, because the inhibition is much more, it seems darker than the inhibition here, because here the shade of this is lighter than this, and it's +5, as opposed to the inhibition here would only be -5. So this is one of a very simplistic ways by which the brain processes color, because it doesn't look at the color. So, I mean, just to, the point being that it is not simply that, it takes each light signal from the visual field, just directly interprets and gives an information. No, it processes the information with respect to a background. So the brain is doing this. And to tell you more that the brain is what is giving you the perception, there is another illusion which I have for you. Just focus on the very black center and focus for about 10 seconds. And then after 10 seconds, as I change the screen, just blink. Now blink. So what did all of you see? I, in front of you, only put a blank screen, but your brain created a reverse image of what you saw in the previous slide. So your eye did not get any visual stimuli or any light from the blank screen, but because you were overstimulated in the previous screen, you got an after image immediately as soon as you blinked your eye. So this is just, an illusion just to say that vision may happen in the eye, but perception happens in the brain. And not just this, your brain can also do, other things. It can do crossmodal interaction. To give you an example, if I ask you which of the two images, from the two images, do you think, to which would you like to give the name Booba, and to which image would you like to give the name Kiki? I'm sure most of you will agree, majority of you would like to ascribe the word Booba to the image here and Kiki to the image on my left, right? This is because what your brain has done is it has extracted this very rounded nature of this, image here and attributed or joined it with the way you pronounce the word Booba. So Booba is also very rounded when you speak, as opposed to Kiki, which requires some emphasis from you. So what it has done is it's extracted the rugged nature of this image and attributed it to Kiki, whereas it has extracted the smoothness of this image and attributed it to Booba. And this was a study conducted where, again, a majority of people attributed this to Booba and this to Kiki. So simply saying that your brain can do cross-modal interaction also. And is there proof for this? Yes, indeed. We have a condition called synesthesia, where there are few people in this condition for whom these alphabets, which to some may look just black and white, to them it looks in colors. And how they discovered this? So if you look at the screen here, and I tell you that within 3 or 4 seconds, count all the number of twos, you may not be able to do it. But a synesthete, or person who has a condition called synesthesia, because for them, they see all the 5s as green and all the 2s as red, they immediately are able to tell you that there are 6 2s in the screen, and that too in the form of a triangle. And is there any, structural basis for this? Indeed, there is a structural basis for this. Just a hint towards the structural basis is that the number area and the color area are very close to each other in the brain. And as I was telling you, if there is even a small slight aberration in the connectivity, and if the connections from one area to the other, they just overflow, and this happens as an aberration during development, what you would have is people who are now able to mix their graphemes or their alphabets or their numbers with the colors and start seeing numbers as colors. And in fact, it is considered that this cross-modal interaction is the key to creativity and abstract thoughts. Richard Feynman, who's a celebrated physicist, saw his equations in a colorful manner. He saw colorful equations. In fact, synesthesia is seen quite commonly in poets, musicians, and painters. And it's even suggested that perhaps the key to Renaissance was that many of the people who were involved in Renaissance were synesthetes. So I would like to, end my talk here and leave you, and perhaps I was able to tell you that how the structure or the birth of the brain, right from the cells to connections, leads to its function, and how this function, particularly the perception, happens in the brain. And with this, I would like to just end with a quote from Oscar Wilde about perception, that if the doors of perception were cleared, everything will appear to us as it is, that is infinite. And this is me. As I told you, I'm a developmental neurobiologist. I recently started my lab at inStem, focusing on studying the development of the cerebral cortex in both mice and humans. And I'll be happy to take questions.
Thank you so much, Bhavana, especially the bit about synesthesia. I got reminded of Shakespeare when in one of his lines in Romeo and Juliet, he says it is east and Juliet is the sun, So,
yes, yes.
And it's one of those beautiful metaphors, and it's possible because of these synapses. So I'm going to hand it over to Raj, who will be doing the questions from the Q&A box, and then afterwards I'll do the questions from YouTube. So people from YouTube, please just stay online while we deal with the questions in the Q&A box for the first 10 minutes or so, and then we'll get to you. So over to you, Raj.
Okay, thanks, Bhavana. That was fantastic. There's some questions from Pushpita Saha, which are very nice. So, is the function of neurons dependent on the position where they're located in the brain? What if the positions are shuffled, or the particular neurons send out similar signals?
Oh, that is a fantastic question, and the answer is yes. The position definitely determines the function, and if you were to shuffle, in fact, we are able to do that. Like, for instance, as I was saying, that if you suddenly, if you the neurons which are in, say, neurons which connect one half of the hemisphere to the other half, right? If instead of making those neurons in the brain, you ask them to you express a factor called FESF2, what happens is these neurons which are to connect from, from this half to that half, they suddenly start making neurons which are actually projecting towards the spinal cord. So you can, if you, even you change some of these molecules, not just the positions, I'll come to the positions, but even if you tinker with the molecules, you overexpress some molecules, suddenly making a different kind of neuron. And that neuron, instead of actually projecting from this to that, it starts projecting towards the spinal cord. So you can do these things. So also there is something called, plasticity in development, that you can do these tinkerings in a certain developmental time window. And in this time window, if you remove neurons from one region to the other, you see that they are still able to retain its original, its original function, which it does. So yes, you can do that. So yes.
Nice. So a couple of questions were on the comparative approach you talked about. So, for example, from Pradeep Gade, he talks about the comparative approach, comparing brains of animals from simple to complex and attributing it to their functions. What do you think are the major roadblocks for such a comparative approach? This leads into another question about the fact that there's no sulci and gyri in the mouse brain. Is the mouse really such a good model for human development? So maybe you can kind of give us a complete answer to that.
Yes. So, so the very basic features, like the ones which I told you about, that how the 6 layers of the cerebral cortex come, some of these features are highly conserved. And because, there is this paucity of getting human fetal tissue, it is difficult to study. And so in those cases where you want to really study the conserved features, the mouse is a very valuable system. You can study all the basic features, like, how the stem cell divides, these things, or how the progenitors divide, or if you remove certain factor from the progenitors, their pool diminishes, and that leads to, a shrunken cortex. Some of these features can be very well studied in the mouse. Having said that, of course, the mouse does not have the gyri and sulci. But that is a very special feature in the human brain. So I think the iPSCs and the organoid models which my lab is also generating are very useful to study some of these features which may not be there in mouse but are present in human. But I think maybe I'm giving a diplomatic answer, but I think each model system is important, because the mouse, it's a live organism. You can do some live experiments. You can look at the entire 3D structure. That is something which is absolutely not possible in the human. So when you want to look at the total, so this is a 3D structure where, as I was saying, the emerging function always comes from the cells, but finally it is the entire organ which needs to function together. So when you want such questions, you need the mouse. But certain features which are different can always be answered from the human.
So someone just from YouTube kind of wants to double check whether gyri and sulci are only there because the body needs to accommodate the huge size of the human brain. Perhaps asking whether there are other functions to the gyri and sulci as well.
I must say that I'm not aware of other functions there, though there are, there are, I think, studies which show that, more gyri sulci could be the reason behind increased cognition. I'll be honest, I'm not aware of such studies. So, I mean, evolutionarily, it seems like it is for accommodating the huge expanse of the brain, but I think we're still going towards understanding what exactly these mean.
Another person from YouTube really asks, does Hydra really have a brain?
It does. There is a nerve terminal. It does. Yeah, I'm not an Hydra expert to either, say that. I think there are other people I think can vouch for it better.
A nice question here is whether there are any occurrences when dendrites can act as axons.
Dendrites connect as axons.
Can act as axons, or whether they can modulate nerve activity.
Yes, so the synapse there is both the pre-synapse and the post-synapse. So whether they can do axon functions, I'm not, I must say, I'm not aware of such a study or something.
Okay.
Yeah.
Okay. So another question, just based on the comparative approach. Why do we still have to use mouse models when we can use brain organoids?
Yeah, I mean, I'm a developmental neurobiologist, so I would say this, that for me, yes, we are going there. I think what we are really doing is we are going towards a model system which is more and more human. But the organoids, the current way in which they are, we are only able to generate small regions. Like I showed them the entire structure where, the structure has the different lobes. Current technology of the organoid, I'm not even sure we don't even know which lobes we are generating, right? So, so if you really want to study brain and make inferences about, forget consciousness, even make inferences with small behaviors, which we do in mouse, you still need a brain which is in entirety has all the structures, and that's not still there in the organoid. So I will say that, with the advent of iPSCs and this organoid technology, the field is moving forward in a very big way. But we really need to see how far do we reach to whether we can completely give up the mouse model. And the mouse model is really useful to study some of these conserved features in great detail, which you cannot do in any organoid, or of course you cannot get human brains.
So following on from that, some neurological disorders are said to be genetically linked.
Yeah.
Can these disorders be traced? Can the pathology of these disorders be traced using mouse or organoid models?
Yes, absolutely. Some of these neurological disorders, I was talking about autism, for instance. In fact, just to give a very recent example, is the Zika virus, which affects neurodevelopment, has been very beautifully chalked out both using a mouse model and an organoid model. So how the Zika virus infects and how it leads to its effect has been very well chalked out. So you can study some of these neurological disorders, particularly which are genetic, because the mouse model system, I think what it offers is the ability that you can do a lot of genetic manipulations in the mouse. And that is very useful, as you said, if you really want to study the genetic basis of this.
So another question was asking about Zika virus, in fact, and
Okay.
I guess it feeds into your work on stem cells in the brain as well. So someone is asking how does Zika virus cause microcephaly?
Right. So Zika virus, so microcephaly is essentially where you have a cortex which is reduced in size. And that is because you are progenitors, or the stem cells, which need to make the entire cortex. If they don't divide well, if they don't proliferate or divide well, you would end up with a small pool of progenitors. And that's why you're not able to, you have a cortex which is shrunken, because there was simply not enough stem cells to make the entire cortex. So what the Zika virus does is these neural stem cells, which are called progenitors, it affects the division of these neural progenitors. And because they are not able to divide well, their numbers are diminished, and that leads to the microcephaly, which is seen in the Zika virus.
Okay. So how does the cerebral cortex and its 6 different layers contribute to plasticity and thought process?
Right. So the plasticity which we discuss is so plasticity is at several different levels, right? So one of the plasticities I was mentioning was a developmental plasticity, which happens only during development and right after birth. So very short window after birth, where you can do manipulations in the cerebral cortex, and it will still be adapt, or it'll still be amenable to this plasticity. But the plasticity also exists at the level of synapses, where you can change the proteins at the synapse, and you may still change the connection or the strength of the connection, and that is the sort of plasticity I think the person is talking about. Right?
Yeah, seems like it.
Yeah.
Someone wants to kind of understand a little bit about the brain-machine interface that Elon Musk's Neuralink is speaking to.
Okay.
What do you think the future is of kind of these neural implants? And is there any similar research going on in India?
I'm not aware of any neural AI interface research in India. And also I must say that I'm also fascinated by this brain interface, and and that's it. I don't know how, I don't, I would say this, that I'm not a computational neuroscientist to comment on it, actually. Yeah.
So I'll ask a combination of 2 questions, and just to wrap this up.
Yeah.
So Yaram Puja asks, how do synapses between neurons cause thoughts, memory, consciousness, and other various functions? And Lisa Changakoti is asking your opinion, being a brain biologist, how would you define the mind?
Okay, so how will I define the mind? Okay, let me answer that first. I think we very well understand where I think I in my slides put where the field is now. When I said that we know from cell to functions like perceptions on all of that. But when it comes to things like mind and consciousness, we still don't, or at least I'm not very well familiarized with those articles or anything. As I said, I'm not a cognitive neuroscience scientist, but there's still many gray areas where we don't understand that how these physical attributes. We do know that the strengths at the synapse, that goes on to determine many things like memory. As one of the question was how memory comes. So the memory, one of the ways memory comes is the strengths at the synapse. That is a very strong determinant of memory. So I think that has a role to play in consciousness, but apart from that, I don't want to hazard any guess because I really don't know in what other ways that this contributes to consciousness.
Yeah.
I think I should also go back and read. I think some of these questions are interesting, definitely. Yeah.
Yeah.
Yeah, I thought the perception itself was so cool. You know, I mean, would you believe that about even in the 60 years back, people thought that vision was very simple. People thought that, you know, the eye is a camera. It's taking a picture. That picture goes to the brain. It just gets inverted, and then that's it. The brain says, this is the picture. But, you know, it's understanding, it's work of so many decades that now we understand that, you know, it's not at all so. It is everything to do with the camera or eye stops at the eye. And it's just electrical impulses out of which, you know, the brain is just simply picking information, color, form, all of that. And that to me was a big fascination. And I thought I really should collect that information and present it. That, you know, a very simple thing, but even the simplest thing is so complex in the brain.
Yeah,
and consciousness, we really, I think we're going there. Yeah.
Yes, it is amazing. Yeah, the amount of computation that's going on, and all of this from these illusions as well. So the amount of information coming from illusions is amazing.
Yeah.
Yeah. Fantastic. Thank you, Bhavana.
Thank you.
Thanks. So I think that's all we have time for questions. Next week's talk is on the limb and bones, and it's from Professor Mitaba Bandapai from IIT Kanpur. So the link for registration is already in the chat box, and I think it will be up in various sites. Chandrakant, do you have anything else to add?
I'm just gonna unveil the poster. Like I'd like to do that for the next talk. Like we did. So yes, this is our poster. We've tried to make it a little bit scary with some skulls and crossbones as well there. So and yeah, so this is yeah. Well, just please register. I put the link in YouTube as well as the chat box here, so everyone can register for it. Please go and register right now. And as usual, it will be streamed. The next talk will be streamed on Facebook, on the Blisk Facebook page. We're doing that for the next session. It will not be there on YouTube immediately, but we'll upload a recording on YouTube afterwards. Because and yes, that's about it from our side. So thank you so much, Bhavana, for
thank you so much, everyone. Thanks a lot for attending. Thank you, Chandrakant, Mahin, and Raj.
Yes, and like we have an insane number of questions which we'll mail to you, and if you want, you can probably take them. We've also Bhavana's compiled a mailing list, compiled a reading list, which we will be sending to everyone who has attended this talk and all the registrants. So we will send it to you very soon once I have that document. So with that, I think this was a wonderful, wonderful session, and I thank you all for coming in such large numbers. Raj, do you have anything to add?
No, nothing. Thank you, everyone. Fantastic questions. I'll try and do a better job of presenting them all next time. Okay.
Great. Okay, closing the call. Thank you very much.
Okay.
Thanks.
Bye.
Bye.